Alright, hi guys, my name is Delvin Lee and welcome to AP Bio 101. Today we're going to be learning about cellular respiration. Cellular respiration begins with glycolysis, an anaerobic process. Glycolysis breaks down one glucose molecule. Glucose enters the cytoplasm of the cell. Two inorganic phosphates from ATP attach to the ends of glucose, resulting in a fructose diphosphate molecule. Because two ATP are spent, this phase is called the energy investment phase. Aldolase, an enzyme, cleaves the fructose diphosphate into two molecules of G3P. This is the end of the energy investment phase. The oxidation of fructose diphosphate releases electrons that are transferred to the NAD+, resulting in NADH. This powers the addition of phosphates to the G3P molecule. Electrons are also added on to the NADH complex. The four phosphates are transferred to four ADP molecules, resulting in four ATP molecules. Since two ATP molecules were used in the energy investment phase and four ATP molecules were used in the energy payoff phase, there's a net gain of two ATP. Other products of glycolysis are two pyruvate molecules and two NADH molecules. After glycolysis, the pyruvate molecule can go into either anaerobic respiration or aerobic respiration. Assuming it's aerobic respiration, the pyruvate will first go into pyruvic oxidation, in which two pyruvate molecules are then transported via active transport into the mitochondria. The carboxyl group from the pyruvate is turned into carbon dioxide, and through some oxidative reactions, one molecule of NAD+ is reduced to NADH. Since there are two molecules of the remaining of the pyruvate is then attached to an enzyme known as coenzyme A, thus making acetyl-CoA. Since there are two molecules of pyruvate, this cycle repeats again, and thus the total products are two molecules of NADH and two molecules of CO2. The two acetyl-CoA molecules are then transported to the Krebs cycle. Acetyl coenzyme A is attached to a 4-carbon molecule oxyl acetate, thus making citrate. Citrate then goes through the oxidative reactions where carbon is removed, making carbon dioxide, and electrons are transformed into NAD+, which gets reduced and becomes NADH. The 5-carbon molecule then gets oxidized where a carbon is removed, making another carbon dioxide molecule, and the electrons are transferred to NAD+, which gets reduced and becomes NADH. A molecule of ATP is also produced. The 4-carbon molecule then gets oxidized, and the electrons are then transferred to FAD+, where it is reduced and becomes FADH2. The 4-carbon molecule then gets oxidized again, and the electrons are then transferred to another NAD+ molecule where it gets reduced to NADH. Oxaloacetate is regenerated and the cycle repeats. By the end of the cycle, 2 molecules of CO2 is made, 1 molecule of FADH2, 1 ATP molecule, and 3 NADH molecules are made. However, since there are 2 acetyl-CoA molecules, this cycle occurs twice, making a total of 4 molecules of CO2, 6 molecules of NADH, 2 molecules of FADH2, and 2 molecules of ATP. After that, the electron carriers NADH and FADH2 are transferred to the electron transport chain. The NADH is oxidized into NAD+ and NADH, and it releases electrons to be transferred to protein complexes such as NADHQ reductase. Using the energy gained from falling electrons, hydrogen ions are actively transported against the concentration gradient over the intermembrane space. The NADHQ reductase then transfers its electrons to a mobile carrier, which carries the electrons to a cytochrome complex. A series of redox reactions within the cytochrome complexes transfers the electrons downhill, releasing energy that helps pump hydrogen ions against the concentration gradient. Electrons are then transferred to the final electron transporter, oxygen, creating water. After that, chemiosmosis occurs. The proton flow back down the concentration gradient, passive transport via the ATP synthase. The ATP synthase uses this rush of protons to phosphorylate ADP and create ATP. And that's the end of aerobic respiration. Now we go into anaerobic respiration, which means no oxygen. No oxygen is in this process. No oxygen. I repeat, no oxygen. We'll first start with alcohol fermentation. First, glucose enters the cytoplasm, and the glucose is cut and converted into 2 G3P molecules, which is then converted into pyruvate. In between the process of G3P converting into pyruvate, 2 NAD molecules are reduced by the oxidation of G3P to make 2 molecules of NADH. 4 ATP molecules are also made, but with only a net gain of 2 ATP, due to the 2 ATP spent in the energy investment phase of glycolysis. Since this is an anaerobic process, oxygen will not be present, and thus oxygen will not be the final electron acceptor. Instead, acetaldehyde will be. The pyruvate molecules release a carbon dioxide molecule, resulting in the change of 3 carbon molecules, 3 carbon molecule pyruvate to a 2 carbon molecule acetaldehyde. NADH is oxidized and acetaldehyde is reduced, causing the NADH to give up its electrons and recycle NADH back to NAD+. The acetaldehyde with the electrons then make ethanol. In alcohol fermentation, the regeneration of NAD+ is used so that the process of glycolysis can be continuous. And now we're going to go into lactic acid fermentation. First, glucose enters the cytoplasm, and then the glucose is cut and converted into 2 G3P molecules, which is then converted into pyruvate. In between the process of G3P converting into pyruvate, 2 NAD molecules are reduced by the oxidation of G3P to make 2 molecules of NADH. 4 ATP molecules are also made, but with only a net gain of 2 ATP due to the 2 ATP spent in the energy investment phase of glycolysis. Since this is an anaerobic process, oxygen will not be present, and thus oxygen will not be the final electron acceptor. Instead, pyruvate will be. NADH is oxidized, and the pyruvate is reduced, causing NADH to give up its electrons and recycle NADH back to NAD+. The pyruvate with electrons is then made into lactate. Thanks for watching my first and last tutorial on anything biology related. Oh yeah, don't forget to check out our study tips.